Hello, my name is Bartolomeo Bosco and I'm a PhD student in the Laboratory of Transcriptional Network at the Center for Integrative Biology of the University of Trento in Italy. As you know, transcription is an extremely complex process involving spatial and temporal organization of transcription factor and cofactor. Most of them, including the human P53, recognize specific cis-acting elements, called response elements, and the binding on this DNA sequencing is required for the modulation of the transcription of target genes. In this work, we would like to show you the protocol to study P53 sequence-specific transactivation functions using Yeast as a model system, exploiting a color reporter gene, or the luciferase, or on the quantification of cell growth, to highlight the main experimental steps and the versatility of these approaches.
Protocol 1. Construction of reporter yeast strains that contain a specific P53 response element upstream of the adenine 2 or the firefly luciferase gene. To construct a reporter strain, first follow steps 1.1 through 1.15 to transform yeast cells with oligonucleotides targeting the desired promoter region. The transformation is based on the standard lithium acetate-based protocol. Once transformants appear on 5FOA plates, usually after 3 days of incubation at 30 degrees, replica plate them onto non-selective YPDA plates and also on YPDA plates containing G418, marking each plate to facilitate their subsequent comparison. Incubate the plates overnight at 30 degrees.